HIV is a small and fragile virus, perhaps surprising considering its huge impact on the world. Inside are 2 strands of RNA, which contain the genetic information for the virus. This is unusual, since most organisms, including humans, store information in DNA. The RNA and viral enzymes are packed in protein to form the core. Surrounding this is a protein matrix covered by a fatty envelope through which project protein spikes. These allow the virus to attach to receptor molecules present on the surface of certain human cells. When HIV is attached to a cell receptor, its envelope fuses with the cell membrane as matrix and core enter the cell. The matrix proteins are lost, leaving the core. Inside the core, the RNA is converted to DNA, and when it reaches the nucleus, the virus DNA enters and inserts itself into the DNA of the cell. This irreversible joining of virus DNA and host cell DNA is called integration and leaves the cell permanently infected. In this integrated form, the virus can remain dormant for long periods, perhaps for years. If the cell becomes activated, RNA copies of the virus are made, which migrate out of the nucleus. Some of these RNA stay intact and can form new virus genetic material. Other RNAs, which have been modified, instruct the cell to produce virus proteins. Near the cell's surface, virus RNA and virus proteins collect together and assemble into new virus particles. These bud from the cell's surface, becoming cloaked in cell membrane. The membrane forms the fatty envelope for the virus. The virus particles mature internally and the life cycle is completed. The new virus particle is now free to infect other cells.
I'll go to the doctors and I get a needle there, just there.
When Eve was born, the doctors didn't expect her to live. She defied the odds, but the blood transfusions that saved her life carried a deadly legacy. 11 years on, she died in her mother's arms.
I'm afraid of dying. Because last year I got told I only had about a few weeks to live because I was so sick.
1,400 children just like Eve are infected every day as the virus continues to cut a swathe across the world. At the beginning of 1999, 30,600,000 people were infected with HIV-AIDS. 1.1 million children, 17.3 million women, and 12.2 million men. It is estimated by the year 2000, 70 million adults will be infected worldwide. Several modification events occur to complete the development of a mature infectious virus. Some of these modifications are carried out by a viral enzyme known as Protease. This enzyme cuts a large precursor protein, known as P55, into 3 smaller protein groups, known as P17, the matrix protein, P24, the core protein, and P7, the ribonuclear protein. These smaller proteins are essential components of the virus particle. If the activity of this enzyme is blocked by a drug, an infectious virus will not be produced, and the virus particle formed cannot infect other cells. Narhex, an Australian company, has been able to develop a series of very active anti-HIV compounds. These compounds have been coded NARDG 35/17. Recent in vitro testing with new anti-HIV Narhex compounds has shown that the newly formed HIV particles are not allowed to mature because the compounds inhibit the buildup of the matrix protein, the core protein, and the ribonuclear protein, producing a virus which is unable to spread from cell to cell. These electron microscopy photographs show the infectious virus characterized by a dark condensed center. Yet in this next photo, the newly produced virus is immature, as can be detected by the lack of the dark center. This is a direct effect of the NARDG 35/17.
Mr. Chris Birch, Senior Scientist, Antiviral Laboratory, states, At this stage, the Narhex DG drug is the most active novel drug synthesised in Australia and examined in this laboratory. The NIH laboratory in the United States confirmed the significant in vitro activity of these compounds. Before the human trial, there was a high level of confidence that the positive results to date would carry through to a successful conclusion. Dr. Chris Birch is leading the preclinical trials.
At Fairfield Hospital, we've shown that the Narhex DG blocker is very active against HIV. Following some minor modifications to the drug, we've shown that the oral bioavailability of this drug in dogs is very good, and this has enabled us to proceed to toxicology studies in 2 animal species. And the results of this shows that even after up to 90 days of oral administration of quite high levels of the drug, there is no adverse side effects in these animals whatsoever. This means that the drug has potential for use in man, and to that extent we've now applied to the Ethics Committee at Fairfield Hospital for permission to treat volunteers for very short periods of time to enable us to gain some understanding of how the drug is going to work in man. And we hope that we'll be able to start these studies in November this year.
The first Phase 1 human trial on an Australian anti-HIV drug at Fairfield Hospital.
Narhex DG 35/17 demonstrated excellent oral bioavailability above the required therapeutic level on 12 people. Dr. Sarah Palmer from the Virology Department at Stanford University reported that she was very impressed with the activity of Narhex DG 35/17 and believed that it would be an excellent drug for patients naive to protease inhibitors, as one of them has the lowest IC50 value of all the protease inhibitors available, the highest requiring 300 times more drug to arrest the virus in vitro. The Narhex team then entered into initial human clinical studies of the promising protease inhibitors, placing them at the forefront of global HIV research and drug development. The location of these Phase 2 studies was Brazil. During the Brazilian Carnivale, HIV awareness risk campaigns are in full swing. Still, this epidemic is a growing problem, as 2,000 new cases are registered every month. In Recife, one laboratory confirms 3 to 6% of its patients a week are HIV positive. It's known how many HIV infections are across the entire country. In this climate of need, Narhex launched Brazil's and Australia's first Phase 2 study of tolerability and efficacy of Narhex DG 35/17 in 12 HIV-infected patients. In Part 1 of the study, 6 patients were given placebos, while 6 other patients received 1,050 milligrams a day of the Narhex trial drug. In Part 2, the patients who received the placebos crossed over to the higher 2,100 milligram per day dose. Part 2 received approval after Part 1 passed all blood chemistry analysis with every major organ function unchanged and no increase in lipids. On day 3 of both parts, plasma samples were collected to correlate pharmacokinetic parameters. On day 6 and 14, samples were collected to determine viral load and CD4 count measurements and safety. The plasma was shipped for viral load to the US and Australia to confirm the laboratory findings in Brazil. The results have sent ripples of excitement around the globe. To put this achievement into proper perspective, one has to appreciate the risk in time and cost, plus the effort required in taking a new drug discovery to a successful therapeutic drug approved for use in humans. Out of 25 multinational pharmaceutical companies that tried to develop an anti-HIV drug, 2/3 failed. The average screening of drug candidates is a ratio of 5,000 to 1 of the drug entering clinical trials successfully. The outcome of the Phase 3 human trials, whilst not certain, enjoys a high scientific expectation of successfully producing an acclaimed registered drug for market. The salient features that make this drug so promising in relation to known drug treatment alternatives are lower toxicity, greater effectiveness due to higher bioavailability, less expense due to a simpler manufacturing process, and potentially less drug required. This may add up to a less expensive treatment. Some of the market leaders require up to 300 times more drug to stop HIV in vitro. When Narhex first embarked on the project, the skeptics gave it a negative probability of success. Leave it to the large multinational pharmaceutical companies, they said. Fortune, however, favours the brave.